need to repeat a bit. Okay, so at the start of DNA replication, okay, so you have a nucleotide activated. Okay, so only those which have been activated will be used later. Okay, so very possibly sometimes, this activation, the addition of a phosphate group, actually sometimes also helps with the recognition by the enzyme. Okay, so your enzymes will only pick something that has been activated. Okay, so and to be incorporated into DNA. Okay. And at the start of a DNA replication, okay, so always a DNA replication begins at somewhere that we call origin of replication. Okay, so we have DNA polymerase. Okay, so FYI, you see the DNA is a super duper, as we learned, each chromosome is a super duper long DNA. Okay, so if you are to, if you are to copy this whole thing, okay, so for sure, you wouldn't just get one DNA polymerase and then, starting from the beginning till the end, that would take too long. Okay, so it's like, for example, if I'm asking you to copy a dictionary, okay, so if I just get one person to copy, it will take forever. So of course, what I'm going to do, I'm going to get multiple person to help me. Everyone starts at different page. Okay, maybe you start at page 1 and then page 101, page 201, and blah blah blah. So all of these spots, okay, the page where you start copying, we call them the origin of replication. Okay, and of course, as we learned, okay, DNA, the two strands are tightly held together. Okay, so for DNA replication to occur, which means to copy those base, okay, you need to separate them. Okay, so for this separation, there's a very important enzyme called helicase. Unwind the helix. Helix. This is the root of the word is helix. Okay, so basically it's just this helicase unwind the two strands. Okay, so where of course in order to separate them, to unwind them or unzip them, you need to break the hydrogen bond. Okay, so somehow this enzyme, okay, yeah, and so with this you form a fork. Okay, so we call this a replication fork. Okay, so yeah, and so it brings about separation of two parental strands. Okay, so basically, long story short, at this stage, okay, your RNA polymerase, and also there's this enzyme helicase which binds to this origin of replication, separate the two strands, okay, by breaking hydrogen bond. Okay, so and then the two strands will be separated. Okay, so they'll be acting as template. Okay, so template, as in, we just look at it and then copy, look at it, copy, look at it, copy. Lagging, and that is lagging, Lagging. Okay, so this is called lagging. Okay, lagging strand, lag behind, and DNA ligase. Okay, GASE. Okay. In SPM, you would have come across the term DNA ligase. Do you remember what does ligase do? Okay, it joins DNA together. Ligate kind of mean like glue. Okay, so anyway, so the two parental strands which have been separated from each other would act as a template. Okay, so of course then what happened is that this free nucleotide is going to come in. Okay, so your free nucleotide coming in. Okay, so maybe, yeah, so you see, so this free nucleotide will come in here, the free nucleotide will come in here. Okay, of course, what nucleotide come really depends on the complete. They have to be complementary base pair. So if this is an A, let's say, then this has to be a T. Okay, so if this is a G, this has to be a C. Okay, so the free nucleotide will be incorporated based on complementary base pairing. Okay, yeah, of course. So they come. So of course, first they will be associated here due to hydrogen bonding. Okay, due to hydrogen bonding. Okay, and then this DNA polymerase is going to catalyze this whole process. Okay, so and also one thing to note is that, okay, one very important thing to note is that DNA polymerase is a very special kind of enzyme in the sense that they are made to only copy a DNA in one direction. Okay, so for example, maybe if I were to ask you to copy all of these text, okay, we are as human, we are geared to just do two parental strand X about, we just go in this direction. Okay, you wouldn't start from here or SDNOBRETSEID because it doesn't make sense. Of course, that's how we work as a human. But in any case, the DNA polymerase, the way it works is quite analogous to how we read things. It would only read things in one direction and also synthesize things in the other direction. Okay, so they always read in the 3' to 5' direction of the template, which means, and at the same time, they would synthesize in 5' to 3' direction. Okay, so this DNA polymerase will only read from 3' in a 3' to 5' direction, and at the same time synthesize in 5' to 3' direction. Okay, so which is why if you look at this diagram, okay, you see that this DNA polymerase, it is moving in this direction because the DNA polymerase is moving in a 3' to 5' direction. Okay, and then synthesize from 5' to 3'. Okay, so at least for this particular strand, so this DNA can keep moving along for a very long time because this is all in 3'. Okay, so and 1 minute. I'll just say, okay, so I'll just keep it simple, my explanation, okay? So it will just keep moving in this direction, okay? But on the other end, okay, so when you, the way for DNA replication work, okay, so you have a replication fork, okay? So your DNA, okay, so if let's say okay, let's think, okay, let's say if I'll probably just do another drawing here. Okay, so let's say if the two DNA, okay, the replication fork, okay, open, okay, so these two strands will be anti-parallel. This is 5' to 3'. So this is 5' to 3'. This, okay, so by the way, what do you describe? Okay, so the yesterday there's a term used to describe the fact that the two strands are what? There's a term anti-parallel. Okay, so the anti-parallel. Okay, so if you see once it's at replicate, so after the helicase bind, they unwind, right? Okay, so you can have your DNA polymerase bind maybe, I don't know, in this direction, and okay, maybe somewhere around here. Okay, let's say, okay. So then what you realize is that, okay, so maybe you bind here and I'll just hold on now. Maybe I'll just do one thing like this. Okay, so let's say this is a DNA polymerase. Okay, so DNA polymerase always read in 3' to 5' direction, which means for this one on the left, it should move in which direction? 3'. So it will move in 3. So it will move in this direction, and then this one will move in this direction. Are we good there? Okay, so they'll move in different direction. Okay, so but then the problem is that this part wouldn't be copied because you open up. Okay, so you see, so of course this is going to move down. So okay, and then it is going to synthesize. So then I will synthesize in 5' to 3'. So as long as move, it will just keep moving this way. Okay, but then for the other one, it will only move in 3' to 5', which means this is the purple one is a newly synthesized. Okay, so this is new strand. Okay, which also means here it hasn't read, right? Okay, so of course this as DNA polymerase continue to move, okay, it will continue to drive the unzipping. Okay, so later this part is going to continue to unzip. Okay, so unzip even further. Okay, so of course maybe okay, so basically it will keep unzipping longer. Okay, so maybe it will further unzip. Okay, so here if you remember, so the one originally on the right, okay, hold on, so 5', okay, so this one, okay, so just now as I said, okay, so this one it will maybe start from here, it will keep going until here. Okay, your DNA polymerase will bind. Okay, so but for the other one, just now we already said it will only move in this direction. Okay, so maybe as you unzip further, then another DNA polymerase will bind so that they would synthesize another strand. Are we good here? Okay, so yeah, okay. So this is a part that can be a bit confusing. Okay, so first you need to remember DNA polymerase can only read and move in the 3' to 5' direction. Okay, so which means when you open, and because of the anti-parallel nature of DNA, okay, you wouldn't have two strands keep synthesizing in one direction. Okay, because they're anti-parallel. So what happens is that you see when I said just now, when this open up, okay, so at least on one strand there is a part which hasn't been copied. Okay, there would be a part which hasn't been copied because it will go this direction, it wouldn't reach here, right? Okay, so what happened is that as the zip further, as the double helix further uncoil, okay, because the unzipping of DNA doesn't, the two strands do not just completely separate, okay, it's like zip, okay, so one part open and then one part, okay, so we slowly open, and then every time when it's newly open, then we copy that part, okay. So, and what happened is that, okay, so for this part that hasn't been read just now, later as it's unzipped further, another DNA polymerase will bind, and then it will form another strand, okay. So, but what happened is these two strands, okay, are not connected, okay. Okay, yeah. These two strands are not connected, so they are fragments, and these fragments has a Japanese name. They are called Okazaki fragments. Okay, so these strands they are called. So the one that is synthesized as fragment, they are called the Okazaki fragments. Okay, they are called the Okazaki fragments. I don't know if it's in your syllabus actually. Yeah, it is there. Okay, they are the Okazaki fragments. Okay, so clearly it is discovered by a guy called Okazaki. Okay, so what you will also realize is that, in this case, you can see that, okay, so for one of the strand, okay, it is it can be kept on reading with just a single DNA polymerase, but on the other one, you need to have multiple, okay, so at least for that section, okay, so it can keep reading for a very long time, okay, and so this one, we say this is because this is synthesized continuously, so we call this a continuous strand. And this one, okay, is synthesized in fragments. It is always slower because you see, when this come here, okay, like I say, this one hasn't been synthesized, okay? So the copying of this part will happen slightly behind later. So we say it lags behind. So this one is called the lagging strand, okay? So you see, here we say the two DNA, okay? Oh, sorry, this is a leading strand, okay? The one that where you see continuous synthesis, we call it the leading strand. The one which is synthesized, okay, the template which is synthesized, read in fragmented fashion, we call it the lagging strand, okay? So later those two strand, okay, will be joined together, okay, by, here this will be done by a DNA ligase, okay? If you remember in SPM, you did learn that, okay, for gene technology, for the cloning, if you want to insert the insulin gene into, you want to stick it, you want to glue it to your plasmid, you use DNA ligase, okay? DNA ligase is like it's like a DNA, okay? It's what we call a molecular glue, okay? So it's basically just glue things together, okay? The DNA ligase, okay? Yeah, so that is what happened there, but during the synthesis of the new strand, okay? So that's what happened, okay? So again, the reason why you have leading strand and lagging strand, two reasons, okay? Firstly, your two DNA strands, they are anti-parallel, okay? Secondly, your DNA polymerase can only read and synthesize in a specific direction, okay? Reading is always from 3' to 5', synthesis is always from 5' to 3', okay? And then at the end of replication, okay, so at the end of replication, okay, so you can just copy this. Okay. So once the replication is done, okay. Okay. So once the replication is done, okay, so the two strand, okay, they're going to rewind, okay, into a double helix, okay. So as I mentioned just now, this whole process is described as being semi-conservative, okay. And at the end, you will produce, okay, at the end of the DNA replication, you have two identical DNA molecules, okay. This one.